Okay, so now we're going to talk about the actual TCA cycle. And I want to remind you about where we are in making energy. So we've already discussed glycolysis, in which we took glucose and we generated pyruvate. And then now we're moving into the step where we're going to generate more energy using the TCA cycle, also known as the citric acid cycle. So we've discussed already how pyruvate dehydrogenase complex will take our 3-carbon pyruvate and generate a 2-carbon acetyl CoA. And this 2-carbon acetyl CoA is going to interact with our 4-carbon oxaloacetate to generate a 6-carbon citrate. And so this is going to be the first part of entering those 2 carbons that came through glycolysis and adding that into the TCA cycle. Now, eventually, we're going to also talk about the electron transport chain in aerobic respiration. So this is just a very big overview of the TCA cycle. And the TCA cycle is going to start with acetyl CoA. And as we mentioned before, acetyl CoA can come from many different sources. We can have acetyl CoA from pyruvate. We can have acetyl CoA from fatty acid oxidation, and from ketone bodies such as acetoacetate. Also amino acids, of course. And all of these are going to generate our 2-carbon acetyl CoA. Now, as you go through the TCA cycle, it's all about following the carbons. We have a 2-carbon acetyl CoA, which is going to combine with our 4-carbon oxaloacetate. This will form a 6-carbon citrate. And then we're going to remove a carbon between isocitrate and alpha-ketoglutarate to form a 5-carbon. And then we're going to remove another carbon between alpha-ketoglutarate and succinate CoA to form our 4-carbon. And then we have our 4 carbons which follow each other back to reset to oxaloacetate. So in the meantime, when we're doing this, we're going to be generating some things that are helpful for making energy. So in the first place, we're going to make NADH, and these can donate their electrons to the electron transport chain. And in addition, we're going to be actually making energy. So you're actually going to generate a GTP, and this will form energy. Okay, so in general, a TCA cycle, around the TCA cycle, will make 1 mole of FADH2, and this will generate 2 moles of ATP, 3 moles of NADH, and you get 3 moles of ATP out of that each, 1 mole of GTP directly, or ATP, but GTP, which is equivalent of ATP, and this is called substrate level phosphorylation because you're making the GTP directly from the enzyme reaction within the TCA cycle. And then each mole of pyruvate that completes the TCA cycle will generate 12 moles of ATP. And remember, our pyruvate came in through the Pyruvate Dehydrogenase complex to make our acetyl CoA. Okay, so here's our TCA cycle from your book, so let's walk through this. We have 8 steps of the TCA cycle. The first step is generation of citrate. And so this is going to be a condensation with acetyl CoA and oxaloacetate. And they are going to use citrate synthase, which is going to form citrate. This has a very negative delta G. And so this thioester bond of acetyl CoA is what's going to help to push this and keep this moving forward. This is also an irreversible step. So once you make citrate, you can't go backwards. This is going to be feed-forward regulated by ADP, and is also going to be feedback regulated negatively by citrate. And so that's that first step that we just did. Now the second step is isocitrate. And so this is going to use an enzyme called aconitase. And aconitase, you're going to have a reversible addition of water to the double bond of your cis aconitate. And this is going to either lead to citrate or isocitrate. Now this is going to favor isocitrate under conditions where you need energy, because in the next step, isocitrate will be rapidly consumed. So here again, you have citrate, which was formed from oxaloacetate, and your acetyl CoA. And then a aconitase is going to generate our isocitrate. Now, one thing to note about aconitase is aconitase has an iron-sulfur center. And so this is important because aconitase moonlights as an iron-sensing enzyme. Now, there are two versions of aconitase. One is going to be in the cytosol, and one is going to be in the mitochondria. And so the one in the mitochondria, of course, is going to be involved in the Tca cycle. But when you have low levels of iron, your iron-sulfur centers are going to collapse. They're going to disassemble because there's not enough iron to hold them together. And when this happens, aconitase becomes an apoaconitase because it lacks this iron-sulfur center. And this is an apoenzyme, which means it's a pre-enzyme. It cannot function. However, apoaconitase is going to bind to messenger RNAs of proteins that are important for iron regulation. So the transferrin receptor and ferritin are going to be bound to by apoaconitase, and they're going to increase the protein synthesis of those two. So you're going to increase transferrin receptor and ferritin. Transferrin receptor is going to bind to iron in the blood, and this is carried by transferrin, and then it's going to allow the iron to enter the cells. And so your apoaconitase is going to regulate iron metabolism through protein synthesis or at the level of translation. Okay, so then after we have our isocitrate, we're going to have oxidation of isocitrate using the isocitrate dehydrogenase complex, and this is going to generate our alpha-ketoglutarate. Now this is an irreversible reaction. This is going to be regulated by ATP. If there's too much ATP, you're going to block this reaction. It's going to be positively pushed forward by a need for energy, either by ADP levels or by calcium levels. So basically, your isocitrate dehydrogenase is going to oxidize your isocitrate and transfer the electrons to NAD. So NAD in the mitochondria. You can also use NADP. And then you're going to get an intermediate oxalosuccinate. And then the oxalosuccinate is going to lose a carbon. So again, remember, citrate came in as a 6-carbon. Isocitrate is a 6-carbon. And so you're losing a carbon here. And so this is going to be our 5-carbon alpha-ketoglutarate. All right, so we started with acetyl CoA and oxaloacetate. We generated citrate. Aconitase made isocitrate. Isocitrate through the dehydrogenase complex lost a carbon and became alpha-ketoglutarate. Now, alpha-ketoglutarate also has a dehydrogenase complex, and this one is going to be similar to the pyruvate dehydrogenase complex. It's going to have an enzyme 1, enzyme 2, and enzyme 3. The difference is in the specificity. So the alpha-ketoglutarate dehydrogenase complex is specific for alpha-ketoglutarate, whereas the PDH complex is going to be specific for your pyruvate. So that means that enzyme 1 is the specificity, so enzyme 1 of pyruvate dehydrogenase will bind pyruvate. The enzyme 1 of your alpha-ketoglutarate dehydrogenase complex binds to alpha-ketoglutarate. But apart from that, they're pretty much the same. So here we have our alpha-ketoglutarate. We have our alpha-ketoglutarate complex. You can see here that we're going to have lipoate, FAD, coenzyme A, NAD is going to be involved in there. And all of this is going to generate succinyl CoA. So again, we're bringing in another coenzyme A, and this will generate a Succinate Coa. This is going to have feedback negative regulation from Succinate Coa, also by NADH, which is generated, so that's another feedback. And then it's going to be pushed forward positively by calcium. Now I should point out here also we're losing a carbon similar to the Pyruvate Dehydrogenase complex, where we brought in a 3-carbon pyruvate and it became a 2-carbon. We're bringing in our 5-carbon alpha-ketoglutarate, and we're leaving with a 4-carbon Succinate Coa. Now, just as a side note, we do have some divergent evolution. Divergent evolution is a case where you have some similarities, that they came from a common ancestor, and then they diverted to have different either specificities or even functions. So in our citric acid cycle or Tca cycle, this is going to be our alpha-ketoglutarate dehydrogenase complex. And so you're going to take alpha-ketoglutarate, and this is going to make Succinate Coa, and it has the enzyme 1, 2, and 3. In our transferring the carbons from glycolysis to the Tca cycle, we have our pyruvate dehydrogenase complex. This is going to take pyruvate and make Acetyl Coa, and again have a series of coenzymes that are very similar, enzyme 1, enzyme 2, enzyme 3. In the future, we'll talk about another dehydrogenase complex, and this is going to be in the oxidation of isoleucine, leucine, and valine, our branched chain amino acids. And then this is going to form an alpha-methylbutyryl coenzyme A, and it's going to have the same coenzymes that are going to be important in this complex as well. So all of these are using the same 5 cofactors we talked about: thymine, pyrophosphate, coenzyme A, lipolate, FAD, NAD. And they're going to have the same kind of complexes, E1, E2, E3. They're all going to be different in the E1 in their specificity. Okay, so now we've made our coenzyme of Succinate Coa. So the next step is the conversion of Succinate Coa to succinate, and this is going to take Succinate Coa synthase. And in this case, we are going to generate a molecule of energy, GTP. And so we're going to, Succinate Coa again has that thioester bond that was similar to our Acetyl Coa. So they have good negative or energy that will push the reactions forward. And so this is what makes this reaction a net negative. And the energy again comes from that thioester bond. And we're going to generate GTP. Now we have succinate, and then succinate is going to donate some hydrogen, some electrons to succinate dehydrogenase. And succinate dehydrogenase is actually part of the electron transport chain. And then after we donate those electrons to the electron transport chain in the form of FAD, which we'll talk about when we talk about the electron transport chain, we are going to generate fumarate. So succinate dehydrogenase has a flavoprotein. It's FAD. And so in this case, we are going to be donating our hydrogens to the FAD in the succinate dehydrogenase complex. And so you can see in this one, it's going to be passing those electrons on to other parts of your enzyme. So this whole thing here is our succinate dehydrogenase complex. And so here is our succinate binding site, and it's going to again pass hydrogens to FAD. And then the FAD is going to pass these on to our iron sulfur centers. So there are going to be 3 of these centers within the succinate dehydrogenase complex. And then in here you also have heme B, which we'll talk about. That's helpful for stabilizing. It actually doesn't do anything with the electrons. And then we're eventually going to transfer these electrons to ubiquinin, which is going to be in the membrane of the inner mitochondria membrane. So This is one of the enzymes that links together the TCA cycle and the electron transport chain. So this is succinate dehydrogenase. So now that we transferred those electrons from succinate to our succinate dehydrogenase and generated our FAD, we now have fumarate. So the next step is to hydrate fumarate to malate, and this is going to occur by fumarase. And so you basically are going to have a carb anion as a transition state, and then eventually you'll generate malate. Now the next step is to oxidize malate to oxaloacetate. So this is the last reaction of the TCA cycle when you go all the way around the circle. It's by malate dehydrogenase. Importantly, this is not a rate-limiting step. This is not a step that cannot be reversed. This step can actually be reversed. And so NAD is going to pick up a hydrogen when you're going from malate to oxaloacetate. And of course, it'll do the reverse if you're going from oxaloacetate to malate. All right, so we have completed our TCA cycle. Congratulations. So on this turn of our TCA cycle, we entered with one acetyl CoA group. We released 2 carbon dioxide, so we started off up here with a 6 carbon, and over here we have a 4 carbon, and so our 6 carbon went around the turns, and we generated again our 4 carbon. So we lost a few carbons. We generated 3 NADH, and all these NADHs go to the TCA cycle. It goes to the electron transport chain. We generated 1 FAD, which is part of the electron transport chain, and 1 GTP or ATP was generated. It could be either GTP or ATP. In this case, we usually talk about it with respect to GTP, but ATP can be generated as well. In addition, we regenerated our 4 carbon oxaloacetate.